Alrighty. So, hello, my name is Marc Villa, and today I'll be presenting my research on combined effect of light and heat on leaf functions. This study was part of the USDA REU program, where I worked under the guidance of Dr. Christine Scafoni alongside grad students Eduardo Barragan and Carly Miller. A little about me. I'm passionate about fungi, and I'm currently studying biology at Cerritos College. Uh, this picture right here shows me holding a coral mushroom at the LA Mushroom Society festival at the LA Arboretum. And these two photos, I also enjoy film photography, so I love taking photos of nature and stuff. And these two photos is just of moss with the sporophytes and then another mushroom. I'm not too sure which type of mushroom that is. uh, okay. Oh, okay. The project was developed during my internship. I collaborated with Eduardo and Carly Miller right here. They're both right here. To collect plant samples from the California Botanical Garden. Our focus was to study how five native California species respond to combined stresses of heat and light. Um, the objective of the research was to determine how light and heat combined impact leaf function in native plants, and to refine methods for studying heat stress in leaves, improving how we evaluate plant resilience to extreme conditions. Um, and as you the with with the globe temperatures rising. They're rising about 1.17 degrees Celsius since the late 19th century. Plants are facing more extreme environmental conditions. In California, average temperatures have increased about 1.8 degrees Fahrenheit, with nighttime temperatures rising the fastest. This highlights the need to better understand how plants handle these changes. And here's a little video of the Earth heat warming up, but I'll let that play. It should be done soon. Yeah, and you can see it pick up around like the 70s with the temperatures increasing, and then from the 70s on, it just gets worse and worse. Mhm. Okay, a little background for the research. Both light and heat stresses impact photosystem 2, a critical protein complex involved in photosynthesis. Stresses increase energy loss of stresses increases energy loss as heat or fluorescence, reducing efficiency. To measure plant health, we use the FE over FM ratio. A healthy plant scores around 0.82. I'm going to be going over that a little bit in the next slide. Um, yeah, to assess the heat sensitivity of plants and stuff, and the native species that we focused on, we calculated the T50, the temperature at which photosystem 2 loses 50% of its efficiency. Um, this was determined using a water bath protocol by Krauss uh, and others. Uh, this was the term allowing us to generate a thermosensitivity curve for each plant species. And on this one, this is the little curve that we're looking at and we focused on during the research. On the x-axis, it shows the temperatures, so 25 degrees Celsius and 45 degrees Celsius, and then our FE over FM ratios on the y-axis. And as you can see, it shows a little that the little line is that T50, the dotted red line. And on these photos, you could see it just shows like what the fluorescence meter uses. This is a little fluorescent meter right here um, on the top right. And the red being that it's healthy, the 0.82, all the red is so it's accepting that photon and converting it into chemical energy. And then the blue spots right here are a little bit more less effective at doing that. Uh, and the five species we studied was the white sage, the Salvia apiana, mountain mahogany, chaparral whitethorn, desert apricot, and summer holly. And uh, we select these species for the ecological importance and their ability to survive in California's diverse and often challenging environments. So we try to choose a little bit from different environments, so like the desert, the chaparral locations and stuff like that. Um, So our first goal was to study the combined effects of light and heat stresses on plant health. To do this, we exposed leaves to 8 to 10 temperature treatments under two conditions: low light, which is like lab light, like normal light, and high light, which we use the LED lights right here on the top left to simulate that the full sunlight. After 24 hours, we measured the recovery using the FE over FM ratio on the chlorophyll fluorometer, and we've calculated T50s for each condition. And then also our second goal was to refine methods for studying plant recovery after the heat stress. To achieve this, we tested three recovery approaches in a recovery in sealed bags with them closed up. Uh, Also recovery in open bags with a damp paper towel inside with all the leaves inside, and then recovery with the leaf petioles placed inside water. And then on these two photos, you can see the one with the leaf petioles placed in the water right there with all the temperatures marked. And the two species that we tested was the Circocarpus and the Salvia. So I believe this one's the Salvia, and this one this is also the salvia as well. I don't have a photo, but okay. And uh, so our results in 4 out of the 5 species studied, the highlight significantly reduced heat tolerance, lowering T50 values by 1 to 6 degrees Celsius compared to low light. This suggests that studies conducted in dark conditions. So in the past, they wouldn't use any highlight; they would just use normal lab light. Um, so the with those studies, they may overestimate the heat tolerance of these plants because they're only focusing on the heat and not light as well. Desert apricot was the exception, showing no difference between light temperatures, and that's going to be right here on the bottom. This was the Prunus fermonti, which is the desert apricot, and that barely showed any difference between the low light and highlight between the confidence interval right here. And the rest, so the salvia, the rest of them, the summer holly, and all of those did show a big difference. So on the y-axis, it shows the temperatures. 30 we did 30 to I believe 54 degrees Celsius, and then the FE over FM on the y-axis. And the white dots are the low light. Oh, sorry. Yeah, the white dots are the low light, and the black dots. Sorry, the black dots are the low light, and the white light dots are the highlight. And then let's keep going. So the results for the refining the methods, recovery methods generally had a little impact on the FE over FM values, except for mountain mahogany, which was the Circocarpus. Um, Open bag recovery led to worse. What we found is the open bag recovery led to worse outcomes, likely due to hydration. This suggests the sealed bags or petioles placed in water are better options for avoiding the water loss. So on this one right here, on the x-axis is the FE over FM again.
1 minute, 1 minute, Mark. One.
Gotcha. The bag and the yellow is the bag, the orange is open, and the blue is water. Okay, and then a little discussion on the findings. Our findings show that the highlight significantly reduces heat tolerance in most species, with T50 values dropping by up to 6 degrees Celsius compared to low light conditions. This suggests studies done in dark and low light settings may overstimulate the plant's ability to handle heat stresses. The reduced tolerance under high light is likely caused by reactive oxygen species, which damage proteins and slow photosystem 2 repair. For recovery, we found that oxygen deprivation had a little effect, but dehydration, especially in open bags, negatively impacted recovery. Seal bags or petioles in water provided the best conditions for recovery, ensuring more accurate results. Um, Overall, the study highlights the importance of examining combined stresses like heat and uh, light and heat, and better understand plant responses in climate and to the changing climate. And we would want to the implications for the future is just to see to have a higher temperature when recovering to stimulate more real world conditions. Uh, and from this internship, I gave me hand-on experience on both lab and field work. I've never done that in the past, so it gave me a chance to see how I liked it and see which one I liked a little bit more. It helped improve my research and public speaking skills and inspired me to pursue a career in
Thank
assessing
you, Mark,
climate
but
change.
we're gonna have to move on. Thank you.
No worries. Thank you.